So Evan Boyar is a systems, software, and electrical engineer with experience in the aerospace industry, looking to transition to a more impactful career. He is also an animal activist and independent researcher, and he's extremely cool. And I'm not just saying that because this is the bio he wrote for me to read to introduce him.
All right, thank you. Once again, thanks for the genuine introduction. So I'm awed by the faculty with which so many people here have presented. I don't have that faculty, so I'm going to be heavily relying on some notes. I hope you don't mind. So many people here and elsewhere think it's extremely likely artificial intelligence, transformative artificial intelligence is coming soon, and will be mainly based on the transformer architecture with perhaps like a few algorithmic improvements or maybe just using it as scaffolding. But basically what we've got right now. I'll admit to being largely sympathetic to these views. However, there is some reason for caution and a possible other means by which we might have rapid progress. You know, weird things sometimes happen. You may be aware. Some of the keener-eyed among you may have noticed the title of this talk. So brain emulation is the less and less hypothetical process of creating a functionally equivalent computational model of a biological brain, usually a specific brain. We've had a surprising pace of development on that front lately. Some of you may have heard about the Open Worm project a while ago. Well, this talk isn't about that because it went more or less nowhere and is based on some stuff that I find pretty questionable. While C. elegans, the worm in question, was first believed to be a cool model organism because it has a tiny invariant nervous system. It turns out smaller brains seem to be actually more challenging to emulate. Each neuron has to do more with complex internal signal processing. Larger brains, as we're finding out, seem to have specialized neurons with clearer, simpler functions. This is according to the lead researcher on a paper that got me very concerned back in October. I recently spoke with him to get a better understanding of where things are going and to ask about his company, which is working on emulating the brains of, for the moment, fruit flies, Drosophila melanogaster. Why would a company be trying to do that? Well, let's detour for a moment and look at the Office of the Director of National Intelligence for the US government, a cabinet-level office. Okay, get ready for some acronyms. It's the government. Their organization, which is a sister to DARPA, IARPA, or the Intelligence Advanced Research Projects Activity, weirdly, launched the Microns program, at least a good name there, Machine Intelligence from Cortical Networks, back in 2016. Their tactic, at least at first, was to focus on mapping at least a segment of the mouse visual cortex, since at that time, visual understanding was something that had thus far largely eluded computational methods. Just this year, the research team on the Microns program announced an amazing feat. The mapping of a cubic millimeter of a mouse's brain. That sounds small, but they managed to get the connectome. That is to say, a model of all the connections between neurons, including the relative connection weights and physical structure, as well as an understanding of the genetic expression of neurotransmitter sites on those neurons and within the brain. 200,000 neurons and 500 million connections. And a mouse brain is fairly small. This was actually a sizable portion. What can be done with this will soon be seen. Their stated goal is, of course, to reverse engineer intelligence from biology. Getting back to the research team I mentioned before, the one with flies, they were also in part funded by IARPA for similar reasons. Their team was working to synthesize some pre-existing data from other teams. The data was somewhat similar to the mouse cortical survey, but in this case, it was an entire fly's brain. One specific fly who was killed for this research. Enter Dr. Xu, the author I mentioned before. His lab essentially took the work of the Flywire group, which, one, created the fly connectome, two, incorporated the work of two other teams to get synapse predictions and guesses at neurotransmitter locations, and three, was once again also funded by IARPA. Notably, Shu's group was then able to create a model that works best for emulating cholinergic neurons, which make up the grand majority of the fly's neurons, and GABAergic neurons, which compose the second largest contingent. It has mixed results for other neurotransmitter sites. However, this was enough to do some pretty incredible stuff. I'll reiterate one fact. This was a whole brain simulation, but most neurons that weren't studied were inactive due to the modeling methods. In any case, by stimulating the emulated sensory neurons, they were able to observe the fly's motor responses. And here's the crazy part. When performing the same stimulation to living biological flies, the flies acted identically to the emulated fly, 95% of the time. That's proof of concept. You can recreate behavior from a connectome. For at least taste, touch, and motion related to taste and touch, there is now an emulated fly's brain. And you can download this individual fly's brain yourself. There's a GitHub. Like, talk about info hazards. Shu now works for a company called Eon Systems, which is, quote, navigating the fastest path to human emulation to safeguard a flourishing future. Intentionally, a second path to AGI. Other research teams are racing to do similar research in zebrafish larvae, mice, other animals, and of course, once again, human brains. Things are moving so quickly that I actually had to add this slide like about a week ago, since more than 100 scientists from around the world just met to kick off a 25-year international project headed by China intended to map primate brains, including human brains, through all stages of life. Iran is spinning up a national primate facility, research facility, meaning animal testing is about to ramp up because of this effort. If you're at all concerned about our future, we must take care to monitor developments in these arenas. If you're worried at all about our future, we must take care to monitor developments once again. If you're worried about the possibility of valenced experience in emulated minds, not merely brains, that is to say, the possibility for those emulations to feel, I hope you'll join me in signing a declaration calling for the recognition of emulated brains as possible moral patients and petitioning that they be granted the same rights that similar biological brains in physical space enjoy. This would allow us, as we grow our protections for animals, for our protections for at least certain types of digital minds to grow in lockstep without any extra effort. Even Dr. Shu agreed extending this kind of care to emulations was prudent. Developments like this are worrying for the effect that they may have on the animals tested on, the emulated themselves, and for their possible role in accelerating AI development. This is all to say, Shu Fly, you're bothering me. If you're interested in learning more about this, signing the petition, checking out the bibliography, learning more about me, and getting in touch, scan the QR code on screen. Now, be well and do good things. Thank you.
Yeah, thank you so much. I wonder what are the stated timelines of these companies to reach kind of human brain emulation, and how what do you think about these stated timelines?
Yeah, so the timelines are kind of all over the place. I've actually in the past few days been able to talk with somebody else who is formerly on one of these research teams. Some people will give fairly short timelines, some will give fairly long timelines. The timelines to emulating portions of the human brain are very short. There are research teams that are probably going to be publishing quite soon. And we already have some small amounts of the human brain connectome mapped. As to full brain, China, at least their consortium, that's a 25-year plan. We might get there faster, we might get there in less time, but I think governments have been pretty good at figuring out how long these things are going to take. But for the smaller brains, yeah, way faster. Yeah.
Thank you.
Mm-hmm. You're welcome.
I think that's all the time we've got. So thank you very much.
Very welcome. Thank you again.